أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. So today we will study about the two species of Clostridium genera, which are Clostridium perfringes and Clostridium difficile. As we have already discussed, a Clostridium genera consists of gram-positive rods which are anaerobic and spore farming. Coming towards the Clostridium perfringes, so it causes two types of diseases, gas gangrene and food poisoning. We will discuss each one of these one by one. Coming toward the first disease, gas gangrene, which is also called myonecrosis and necrotizing fasciitis or flesh eating. So what is the epidemiology of the Clostridium perfringes causing the gas gangrene? So the spores of Clostridium perfringes are located in the soil which enter the body through war wound and road traffic accidents. Whenever there is a wound in the war or an RTA, the spores enter the wound. The gas gangrene caused by Clostridium perfringes is also associated with the endometritis. Also, the vegetative cells of Clostridium perfringes are the member of normal flora of colon and vagina. How it causes the gas gangrene? What is the pathogenesis of the gas gangrene caused by Clostridium perfringes? So, when the Clostridium perfringes enter the traumatized tissue during war wound or a road traffic accident, so it produces toxins, which are alpha toxin, also called lysithinase, and degradative enzymes. The alpha toxin damages the cell membrane of all cells, causing tissue injury and also cause hemolysis of the RBC, red blood cells. While degradative enzymes produce gas like hydrogen in the tissue. Coming towards the clinical finding of gas gangrene. Gas gangrene. So there will be pain at the local site, there will be edema, there will be necrosis in the wound area. In on palpation, if there is a cracking sound called crepitus, then it shows that hydrogen is produced by anaerobic bacteria. Other clinical findings are hemolysis due to the alpha toxin, jaundice due to the hemolysis of RBC produced by alpha toxin. There are also blood-thin exudates in many areas, and as we have discussed that the gas gangrene is also associated with the endometritis or septic abortion, so there will be foul-smelling bloody vaginal discharge, and if untreated it can cause shock and death. This is the picture of gas gangrene caused by Clostridium perfringens. So coming towards the other disease, which is food poisoning, the epidemiology of the Clostridium perfringens causing food poisoning is that the spores in the soil contaminate the food, and these spores are resistant to heat, so they can't be eliminated by inadequate cooking. But remember that the vegetative cells Clostridium perfringens are the member of the normal flora of colon, but not the small intestine. So whenever it enters the small intestine, it causes food poisoning. The pathogenesis of food poisoning is that the Clostridium perfringens produced an exotoxin called enterotoxin, which is a super antigen producing interleukin 1 and 2 from the macrophages, neutrophil, etc. This interleukin 1 and 2 act on the enteric nervous system, causing vomiting and diarrhea. Coming towards the clinical finding, so the food poisoning have incubation period of 4 to 8 hours and characterized by the watery diarrhea, cramps in the abdomen, and vomiting, but little vomiting, which resolves within 24 hours. So the laboratory diagnosis for Clostridium perfringens causing food poisoning is usually not done, and the treatment is symptomatic treatment. But the laboratory diagnosis for the disease gas gangrene or necrotizing fasciitis caused by the Clostridium perfringens is done. So it is through microscopy, culture, and biochemical tests. Microscopy, we can see gram-positive rods, and the spores can't be seen due to the large amount of nutrients in the smear. On culture, we do two types of culture: blood agar culture and the egg yolk agar. The blood agar culture shows characteristic double zone of hemolysis, which you will see in the picture in the next slide, and the egg yolk agar on which they are cultured anaerobically, it produces a precipitate in egg yolk agar due to the enzyme lecithinase. Biochemically, they can ferment sugar and they produced organic acid. And the treatment is penicillin G. Now coming towards the other species, Clostridium difficile. So it is a gram-positive broad anaerobe pore forming, as we have also already discussed, and it is the most common nosocomial cause of diarrhea. Coming towards the epidemiology, so it colonized the large intestine of about 3% of the general population and 30% of the hospitalized patient. And these people, it lives as a normal flora and can be transmitted through eco-oral route by either spore or the bacteria itself. Coming towards the pathogenesis, it rarely invades the intestinal mucosa. Pathogenesis is antibiotic associated. It means that antibiotics suppress drug-sensitive members of the normal flora, which allow the Clostridium difficile to grow and produce exotoxin A and B, which are both glucosyl transferase. Exotoxin B playing the main role in the disease. So exotoxin A and B both glucosylate G protein, which is also called pro-GTPase. It then in turn causes depolymerization of actin, which causes loss of the cytoskeletal integrity, apoptosis, and in turn death of the enterocytes. Antibiotic which cause antibiotic associated diarrhea are the clindamycin. It is the first to be known, third generation cephalosporin, ampicillin, fluoroquinolones, drugs used in the cancer chemotherapy, and the proton pump inhibitor. Coming towards the clinical finding, it causes pseudomembrane colitis, which is a disease characterized by diarrhea which is associated with a pseudomembrane. This pseudomembrane is a yellow-white plaque on colon of mucosa. Diarrhea is usually non-bloody and the neutrophils are found in stool. It also causes toxic megacolon, fever, and abdominal pain. Coming towards the lab diagnosis, so the culture and microscopy of the snare are not suitable because they may be the part of normal flora. So we use two kinds of tests or diagnosis. One is to detect exotoxin by the ELISA and the other is to detect genes that encode the exotoxin by using polymerase chain reaction. Coming towards the treatment, so the treatment is to withdraw the causative antibiotic, fluid replacements, and by giving oral metronidazole or vancomycin, which is usually given in severe cases.